Two fishermen, same boat, same time, same lure. One of them left with a full cooler, snook, redfish, a huge flounder. The other left with zero. Nothing. Not a single bite. Do you know what separated them? An ingredient that costs less than $1, and that you probably have at home right now. The fishing industry in the United States makes billions a year selling synthetic attractants, miracle sprays, imported gels, and none of them come close to what the old fishermen of the Florida, Louisiana, and Texas coasts have known for decades. If this is your first time here, subscribe and activate the bell, because what we teach here is exactly what they don't want you to learn. It was an October morning, Clearwater Beach, Florida. Crystal clear water, rising tide, perfect conditions. Two guys decided to do a real test. Not a lab test, not a paper test. A real test, with a fishing hook in the water. The first fisherman used pure artificial bait. New soft plastic, shrimp-colored, the model that was in the display case for $12.99. Impeccable technique, accurate casts, well-paced retrieval. First fishing spot? Nothing. Changed location? Nothing new. Tried again. 3 weak strikes in 2 hours. Lost all 3. Left the fishing spot with a story of, "It was difficult today." The second fisherman used the same bait. Same model, same color. But before casting, he rubbed the bait on something. A dark, strong-smelling paste that he prepared at home and kept in a small jar, the kind you use to store condiments. Total cost, cents. First half hour, 4 snook. 2 of them over 24 inches. Third half hour, 2 redfish, and a flounder of almost 4 pounds. End of the day? 23 fish against 3 bites that didn't result in anything. Same bait, same boat, same location, the same tide passing by both. The whole difference was in that little pot. Now you might be thinking, but it was a coincidence. The fish were concentrated in that spot. That explains why, when the two switched positions in the boat, the result remained the same. The second fisherman kept catching fish. The first kept waiting. This isn't luck. It's not a secret spot. It's not superior technical skill. It's chemistry. It's biology. It's what the ancients knew and what the industry has spent decades trying to bury under shelves full of expensive products. And the most ironic thing? The synthetic attractant the first fisherman used cost $18.99 at a fishing lodge in Tampa. The second fisherman's paste cost practically nothing. Have you ever suspected something was wrong with that? Because there is. And that's exactly what we're going to talk about today. Before we continue, I need to tell you something. This little jar trick is just the tip of the iceberg. There's a whole universe of knowledge that traditional fishermen mastered: reading the water, positioning, fish behavior, timing with the tide, that the industry prefers you never learn. Why? Because fishermen who know these things stop spending money on products. It's that simple. I gathered all of this together in high-level fishing, the water game. This isn't a theory book. It's a straightforward field guide where you'll learn how to identify productive zones in seconds just by reading the water's surface. Where saltwater fish actually are based on underwater structure, reefs, seagrass meadows, drop-offs, channels, how currents and tides act as an invisible engine that moves the fish and that you can use to your advantage, how to position yourself and attack at the right angle, which often determines more than the lure itself, the behavioral patterns that allow you to predict where the fish will be before you even arrive, and the mindset that separates those who try from those who decide. All in one place, no fluff. The link is in the description. Now, let's get to what you came here to see. The ingredient is Raw squid marinated in coarse salt. Wait, before closing the video, listen to the reasoning. It seems too simple, I know. But that's exactly what the old fishermen of the Gulf of Mexico used. On the Louisiana coast, where redfish is king, the river dwellers of the Mississippi Delta would macerate pieces of squid with salt and let the mixture sit for hours before going out to fish. It wasn't intuition, it was results. Generation after generation, the knowledge was passed down like this. You catch the squid, you macerate it with salt, you wait, you catch more. On the Florida coast, especially in the Tampa Bay and Charlotte Harbor region, two of the best estuaries in the American Southeast, old fishermen did exactly the same thing, except that they sometimes added a touch of crushed dried shrimp. The result was a paste with an intense, almost aggressive smell, which they simply called the dough. In Texas, in the shallow waters of Galveston and Laguna Madre, this practice existed with slight regional variations. Sometimes it was squid with salt and shellfish juice. Sometimes it was just very well mashed, almost liquefied squid. The base was always the same. The salt opens the squid's fibers and forces out an absurd amount of organic compounds that would normally remain trapped inside the tissue. And why did this fade into oblivion? Because in the 1970s, the first industrial attractants arrived in the United States. Colorful little bottles with a synthetic fish scent. The industry campaigned heavily, paid for advertising space in fishing magazines, sponsored tournaments, and placed these products in the hands of the most famous guides in Florida. In 10 years, the collective memory of fishermen was replaced by loyalty to the off-the-shelf product. The old ones died, the young ones bought them, and the knowledge was slowly lost, one generation at a time. But science has never changed. The fish hasn't changed. What works today is the same as what worked 60 years ago. Now comes the part that changes everything. The explanation of why this mixture works in a way that no $18.99 spray can imitate. Saltwater fish have a chemosensory system that puts any other marine animal to shame. The snook, for example, one of the most sought-after fish in Florida waters, possesses two independent olfactory systems working simultaneously. The main system detects compounds in concentrations on the order of parts per trillion. This means it can smell a food source from distances that would make any human hunter blush with envy. When you macerate raw squid with coarse salt and let the mixture rest for the appropriate amount of time, a process called cell lysis occurs. The squid muscle cells rupture under the combined action of the mechanical pressure of maceration and the osmosis of the salt. This releases a huge amount of compounds that are normally encapsulated within the cells out of the tissue. Which compounds? The main ones are free amino acids, especially taurine, glycine, alanine, and betaine, as well as nucleotides such as inosine monophosphate and small concentrations of bioactive peptides. These compounds are exactly what saltwater fish have evolved to detect as a sign of high-quality food. Think of it this way. It's like walking down any street, and suddenly the smell of fried bacon wafts past your nose. Your stomach reacts before your brain processes anything consciously. It's automatic. It's instinct. For snook, redfish, flounder, and striped bass in the American Northeast, those amino acids released by macerated squid are the equivalent of the smell of bacon. Their brains translate that chemical signal as high-energy prey available now. And the hunting instinct kicks in. Taurine, in particular, is an amino acid that exists in very high concentrations in squid muscle, reaching 40 millimoles per liter in fresh tissue. When you macerate and release this taurine into the aquatic environment, it diffuses in a chemical plume that can extend for tens of meters depending on the current. The fish traces this plume back to its source. It's not magic. It's pure biochemistry. Synthetic attractants attempt to mimic this with artificial compounds, usually a mix of synthetic amino acids and processed fish oils. The problem is that industrial processing destroys part of the molecular structure of these compounds. What ends up in the bottle you buy is already a degraded version of what naturally exists in fresh, macerated squid. Furthermore, the compound profile is simplified. They try to reproduce 2 or 3 main amino acids, while macerated squid naturally releases a cocktail of more than 20 different compounds. The fish's olfactory system detects the difference, not consciously, but behaviorally. The response to a natural and complex chemical signal is stronger, faster, and more consistent than the response to a simplified and artificial signal. This also explains why the same lure works better with macerate than without it. The lure alone appeals to sight and the lateral line system, vibration, movement. With macerated squid, you add the olfactory and gustatory vectors. When the fish touches the lure for the first time, even if it's an exploratory touch, it feels the chemical compound on its palate, and instinct tells it to swallow. Without this, an exploratory touch often ends in rejection. With this, the touch becomes a bite. Now, the step-by-step instructions. It needs to be done the right way for it to work. You will need 1 medium-sized fresh squid, the kind sold in Asian markets or supermarkets. They cost between $2 and $3 per squid, but you'll only use half. 2 tablespoons of kosher coarse salt or sea salt. Avoid iodized salt. Iodine can interfere with the active compounds. 1 teaspoon of crushed dried shrimp. Optional, but enhances the result, found in Asian or Latin American markets for less than $2 a package. 1 small glass container with a lid, like a canning jar. Step 1. Clean the squid, remove the outer purple skin and beak, but keep everything else, including the tentacles and mantle. Many people throw the tentacles away. Mistake. The tentacles have the highest concentration of taurine per gram of tissue. Step 2. Cut everything into small pieces, about 2 centimeters at most. The smaller the piece, the greater the surface area in contact with the salt, and the faster the cell lysis. Step 3. Place the pieces in the glass jar. Add the coarse salt on top and mix well with a fork. You want the salt to coat all the pieces evenly. Step 4. Cover the jar and let it rest. Here's the secret that most people get wrong. The minimum time is 4 hours. Ideally, it should be 12 hours. Leave it overnight to use in the morning, or prepare it in the morning to use in the late afternoon. Less than 4 hours, and the lysis process hasn't gone far enough. The squid will be macerated on the outside, but the inside of the muscle fibers hasn't yet released the most important compounds. Step 5. After resting, you will notice that a dark, viscous liquid has formed at the bottom of the jar. This is the macerat. It's where the highest concentration of amino acids is. Don't discard this liquid. It's the gold of the recipe. The ideal point: the squid pieces should be tender, almost falling apart, with a grayish color. The smell will be strong and characteristic, similar to the smell of the bottom of a professional fisherman's boat. If it has a very pronounced ammonia smell, the squid was old. Discard it and start with fresher product. Storage. In the refrigerator, it lasts 2 days without losing effectiveness. In the freezer, it lasts up to 3 weeks. But upon thawing, the lysis process restarts, and the macerat becomes even more intense. Some Texas fishermen intentionally freeze it for 24 hours before use precisely for this reason. Total cost of a batch that lasts 2 or 3 fishing trips? Less than $4. The over-the-counter synthetic attractant that promises the same? It ranges from $12 to $25 depending on the brand, and you'll use an entire container on one long fishing trip. Now, if you want to boost the macerate even further, there are 3 additions that make a real difference. Dried crushed shrimp. I've mentioned this before, but it's worth detailing. Dried shrimp is naturally rich in betaine, a compound that very specifically activates the taste receptors of various saltwater species, especially flounder and pompano. Add a teaspoon per serving of squid and mix it in with the salt. The result covers a broader chemical profile. Sardine oil. Half a teaspoon of pure sardine oil, not olive oil, but the specific sardine oil you find in supplement stores, added to the already prepared bait, changes the visual dynamics of the lure in the water. The oil expands in thin layers on the surface, creating a visible plume of oil that attracts fish from above as well, not just by smell. Especially useful for snook in weak current conditions. Blue crab extract. If you have access to fresh blue crab, and on the American East Coast that's not difficult, grinding the legs and adding the juice to the macerat creates a specific amino acid profile for striped bass and black drum. These fish respond to N-acetyl-D-glucosamine, a compound that exists in high concentration in the crab's exoskeleton and which perfectly complements the squid's profile. With these 3 additions, your bait now works on 4 fronts simultaneously. Smell with the amino acid plume of squid, taste with the betaine of shrimp, sight with the trail of sardine oil, and a specific crab profile that triggers the attack instinct for the most difficult species. And then comes the obvious question. If it works so well, why haven't fishing stores ever taught you this? The answer is uncomfortable but straightforward. Because there's no profit margin in teaching you how to use squid and coarse salt. The attractant industry in the United States generates over $300 million a year. Do you think this market survives if fishermen learn that the best alternative costs less than $4 and lasts for several fishing trips? It won't survive. Manufacturers of synthetic attractants invest heavily in sponsoring tournaments. You go to fishing championships in Florida, and the logo of the industrial attractant is everywhere. On the guide's shorts, on the event banner, on the prize packaging. This is not by accident. It's calculated brand positioning to associate the product with the idea of success and professionalism. When you see your favorite guide using Spray X, the subliminal message is, if he uses it, it's because it works, and it's the best there is. But the guides who fish for a living for real, not the ones on TV, the ones with small boats who go out every day, most of them use homemade bait or something similar. Ask an old inshore guide in Southwest Florida what he puts on his bait before he goes out. Some will tell you. Many won't, because it's their advantage over the competition. This is what happens when knowledge becomes a commodity. What was once passed down freely, from father to son, from guide to apprentice, at the end of the day after fishing, has been replaced by a shelf full of products with attractive packaging and promises of guaranteed results. And the fisherman wasn't the one who benefited. Now, let's talk about how to use it in practice. Because there's no point in having the perfect macerate if you use it incorrectly. Proper application begins before assembling the lure. With a small brush, like a culinary brush, or even a piece of foam wrapped around a string, cover the artificial lure with a thin layer of macerate. It doesn't need to be soaked. A thin, even layer is sufficient. Excess can run off and create a very concentrated plume near the hook, which paradoxically can make the fish hesitate. Too much stimulation at once activates distrust in some species, especially snook in clear water. The timing between application and release, a maximum of 30 seconds. If you delay too long, evaporation and dilution in the air will have already reduced the concentration of the compounds on the surface of the bait. After every 2 or 3 applications, reapply. The compounds dilute quickly in the water. The effectiveness of the first application lasts on average 3 to 4 minutes of active application. In still water, anchored in a channel, the effectiveness time is slightly longer because the plume forms and remains close. In strong currents, you need to reapply more frequently. The best times? The first hour after sunrise and the last hour before sunset. During these periods, saltwater fish are in active feeding mode, and their olfactory system is being used more to locate prey than at any other time of day. Incoming tide is generally better than outgoing tide for snook and redfish in estuaries. The fish move upstream to the seagrass meadows and are actively hunting. Water temperature makes a difference. Below 60 degrees, 15 degrees Celsius, the fish's metabolism slows down, and the response to the attractant becomes slower. It still works, but you need more patience. Leave the bait still longer in a current, and let the chemical plume do its work. Above 80 degrees, 27 degrees Celsius, the fish become more active but also more selective, preferring targets that release a strong signal quickly, which is exactly what Macerat provides with its concentration of amino acids. For snook in tidal passages, those channel mouths where water flows between mangrove islands, position the lure on the downcurrent side of the passage. The current will carry the chemical plume across the entire passage. Snookus that are using the current to conserve energy and capture prey will detect the plume before seeing the lure. By the time the lure passes their location, their instinct has already been activated. For redfish in shallow water, especially when you see their tails cutting the surface as they root in the mud, searching for crabs and shrimp, here, Macerate with added crab extract is devastating. The redfish is head down, focused on smell, and a crab-smelling bait that appears in front of it will be attacked before it even sees properly. For flounder, which stays still on the bottom waiting for prey to pass overhead, cast a little beyond where you suspect it is, and present slowly, practically dragging it along the bottom. The chemical plume will descend with the lure and create a horizontal trail that the flounder detects with its lateral line receptors and sense of smell simultaneously. This is one of the easiest fish to catch with Macerat because it literally cannot ignore the stimulus. Striped bass in the Northeast, Massachusetts, Connecticut, Rhode Island respond very well when you add blue crab extract to the Macerat. The striper uses shallow inlets and estuaries of coastal rivers during its fall migration and is in aggressive feeding mode. The combined signal of squid and crab is one of the most effective combinations that exists for this species in cold water. A common mistake is using excessive macerate, believing more is always better. A fish's olfactory system functions best when it detects a gradient, increasing concentration as it approaches the source. If you saturate the area with excessive macerate, the gradient disappears and the fish loses its directional reference. It detects the smell everywhere but can't trace the origin. The result? Confused fish, hesitant bites, and lower strike conversion. Use sparingly, and frequent reapplication is more effective than using too much at once. Think about your next fishing trip. While the guy on the boat next to you leaves home with the $20 spray he bought at Bass Pro and will need to use the whole can for the entire day, you leave with a small glass jar that costs less than a coffee and has 2 weeks' worth of fishing left inside. In 30 minutes on the water, you already understand the difference. The bites come more frequently. The strikes are more decisive. The fish swallows deeply because their taste confirms what their sense of smell promised. The cooler keeps filling up while your neighbor is still waiting for the first bite. Your boatmates will ask what you're putting on the bait. You decide what to do with that advantage. What the industry spent decades selling for a fortune is now in your kitchen for practically nothing. Squid. Coarse salt. 4 hours of rest. That's all that separates an ordinary fishing trip from one that will become a story to tell. Fishing isn't about luck. It never has been. The old Gulf guides knew that. The river dwellers of the Mississippi Delta knew that. The shallow water masters of Charlotte Harbor knew that. The knowledge was there. The industry just made sure you never stopped to ask why. And if you want the rest, water reading, positioning, fish behavior, the mindset of someone who's really good at fishing, the high-level fishing, the water game, it's in the link in the description. It's the complete knowledge they tried to bury. Now in your hands. Subscribe to the channel and turn on notifications. Next week, there's more stuff they don't want you to know.